Thanks everyone for joining us here tonight. I'm going to give you a short presentation on some recent work we completed on some Ontario beef cattle farms. Um, so the title of the presentation is Beef Farm Management and Pathogens, What the Research Shows. And so I'm sure everyone on this call is familiar with different ways of grazing cattle. And so typically we see in Ontario people that graze conventionally or rotationally, where cattle are moving through defined paddocks for different durations of time. And so in our study, we were looking at rotational farms that grazed between 1 and 7 days before moving from one paddock to another, as compared to farms that continuously or conventionally graze just kind of in one large paddock. And so we were a bit interested in this work because there has been some there's some evidence that rotational grazing can contribute to improved soil and vegetation in terms of soil organic content, nitrogen fixing capabilities, plant distribution, and reducing spot grazing of livestock on land. There's a little bit of evidence in terms of improvements in water quality, in terms of stream bank stability, and contamination of microorganisms making their way into adjacent waterways. Now, there's very little research right now on the impact on cattle health. So there's a bit on nutritional quality, and we found one paper potentially on pathogen exposure, where rotational grazing might reduce pathogen exposure to cattle. And so this led us to develop some research questions. And so what we were trying to do is look at whether rotational grazing might improve soil health while also improving animal, environmental, and human health by one, reducing the presence and quantities of pathogens in livestock, and then also reducing pathogens and antimicrobial resistant pathogens in receiving environments, which we defined as water, soil, and wildlife in this project. And so this is governed a bit by some hypotheses that we developed in studying the different grazing practices. And so when we look at conventional grazing, we typically see kind of concentrations of fecal material on land where the cattle like to hang out, essentially. And then in rotational farms, we see this more even distribution of fecal matter on the field because the cattle are forced to graze in these different sections, and the fecal material tends to be more evenly distributed. And so we hypothesized that perhaps on conventional farms, we might see more runoff of fecal material and subsequently pathogens that are harmful to animal and human health, making their way to surface water and to groundwater. And then we assumed that it might be less on rotational farms. We also hypothesized that perhaps cattle might be interacting with each other's feces more regularly in conventional farms compared to rotational farms, where cattle will be moved into a paddock where they haven't been in a very long time, and therefore that fecal material will have a lot less pathogens in it. We also hypothesized that perhaps we might see impact on wildlife that reside in close proximity to the different farm types. And so I want to acknowledge that this project involved a lot of different collaborators. And so Dr. Charlotte Winder was our ruminant vet on the project. Claire Jardine is a wildlife vet who was involved in the project. Dr. Carrie Dunfield is a soil microbiologist. Dr. Nicole Ricker is an antimicrobial resistance expert. And I wear a bit of an engineer, public health, water microbiologist hat. And then we also had great producers that were involved and also helped us with some of the study design at the outset. So in terms of pathogens of interest, when I talk about pathogens, I'm talking about bacteria and protozoa that can cause illness in human, but also illness in young livestock. And they're zoonotic, meaning they can be transmitted between humans to animals and back from animals to humans. So we were particularly interested in pathogenic strains of E. coli, Salmonella, Campylobacter, and then also protozoa, we were looking at Cryptosporidium and Giardia. In terms of our methods, we wanted to compare the presence of pathogens across soil, fecal, water, and wildlife samples. We also conducted a detailed questionnaire on farm to document animal health, feed, and different management practices. And we also collected rainfall data by installing rain gauges at all the farms. We started the study in 2022 and it went through 2024. We sampled at 10 farms during 3 grazing seasons. And over that time, we collected 648 soil samples, 515 fecal samples, 84 surface water samples, 103 groundwater samples, and 65 passive water samples. And essentially, we went to the farms every 2 to 4 weeks, depending on which grazing season we were looking at. And so for soil and fecal sample collection, we collected 5 unique fecal samples that were freshly defecated, like when we were on site, from both different farm types. We also collected soil samples along transects. So we would collect 3 soil samples across 3 transects where cattle were grazing when we arrived on farm for conventional farms, and then in an area where they weren't grazing to try and represent the different exposure of the cattle to their soil environments. We did the same on the rotational farms where we would transect the field and collect soil samples where the cattle were grazing or in a paddock that they just finished grazing in, and then also in a paddock they were about to move into to also characterize that exposure. We also collected water samples. So we collected 1 liter grab samples from groundwater or well water and surface water sources and analyzed them for what we call fecal indicator organisms. So E. coli, total coliforms, and fecal enterococci, which are indicators of microbial contamination. But we also concentrated large volumes of water on site. So for surface water, we concentrated 500 liters of water on what we call an ultrafilter membrane, which I'm showing in my picture here on the right. Or for groundwater or well water, we concentrated 800 liters of water. And essentially, we take that filter back to the lab and back flush it. And then concentrate that sample down so we can look for pathogens. We also deployed what we call passive samplers, which are these little torpedo boats with their little membrane filters inside. And we deployed those to look for contamination in between our sampling events. So these would sit in the water for 2 weeks to a month and hopeful to try and capture kind of intermittent contamination that might occur when we're not sampling. We also did small wildlife trapping on the farms, and I won't present those results today because they're still in progress. So this work was largely done by Carly Dinga and Sejin Cheong, who have just finished working with me, and they might be listening in today, but they were the ones that compiled a lot of the results of this work, so I want to give them credit. And so I'm going to try and walk you through the results. I'm not going to show you a pile of statistics. I mean, we created kind of a heat map to try and summarize what we were finding, and so I'm going to try and walk you through this, so hopefully you can follow me. So in this table, we've got the different types of samples we collected. So we collected soil, fecal samples, groundwater or well water, and surface water samples. And then all the colors represent a different pathogen that we found. And so blue is Cryptosporidium, green is Giardia, pink is Salmonella, peach is pathogenic E. coli, yellow is antimicrobial resistant E. coli, purple is fecal enterococci, and then the red is Shiga toxin producing gene, which is associated with pathogenic E. coli. And so you can see here, total number of animals that a particular farm had was really important for the presence of pathogens in fecal samples. And this was regardless of rotational versus conventional farm. Stocking density became quite important on conventional farms. So that's what this C means for Cryptosporidium and across both farm types in terms of fecal samples. We also saw stocking density was influential in surface water contamination for Giardia. Next, we looked at paddock size and total pasture size. So paddock size would have been the typical size for a rotational farm that the cattle were grazing in, or often the entire pasture for a conventional farm. And we can see that paddock size influenced the presence of Cryptosporidium. So the larger the paddock, the less Cryptosporidium we saw, and the same for enterococci in our fecal samples. We saw the same relationship with total pasture size. So essentially, the larger the farm, the less we saw in the fecal samples of antimicrobial resistant E. coli and enterococci. We also looked at number of days grazing. So the longer the grazing season seemed to impact the presence of pathogens across soil, fecal, groundwater, and surface water, particularly Cryptosporidium. Paddock resting days, it was interesting that seemed to influence contamination reaching groundwater supplies. So we saw Giardia, Cryptosporidium, and Salmonella in groundwater, and this seemed to be related to the number of days that the paddock was resting over time. So in the rotational farms, we see a lot of resting, whereas the conventional, we don't see any resting period. In terms of number of days in a paddock, this is pretty unique to rotational farms in terms of soil. We saw an impact. And then for conventional farms, the number of days in paddock is usually the entire grazing season. So the longer the number of days in paddock, we saw an impact on groundwater for Cryptosporidium and Salmonella. We also looked at supplemental feed, so whether the cattle were fed hay or natural feed, and we saw a relationship with pathogenic E. coli and enterococci. When cattle were fed hay or natural feed, we saw a reduction in fecal counts of these organisms. We also looked at kind of some environmental factors, so soil moisture and precipitation, and these influence the presence of pathogens across the different sample matrices, which is not surprising. And lastly, we looked at deworming, which seemed to impact the cattle microbiome, in our opinion. And so we're seeing with deworming, we're seeing reduced antimicrobial resistance E. coli, but then we seem to be seeing maybe more pathogenic E. coli. We also saw that when antibiotics were used before a sick animal was isolated, we saw an increase in antimicrobial resistant E. coli in those fecal samples. And we found that farms that applied manure on pasture had higher concentrations of fecal enterococci in their cattle fecal samples. So what does this all mean? I've kind of tried to distill it down into some key findings. In terms of pathogens found in our cattle, as you could kind of see, the pathogen shedding by cattle is complex, and the factors really seem to be interrelated. But the factors that do seem to influence pathogen shedding is the total number of animals on farm, cattle age, total pasture size, stocking density, paddock size, total number of days grazing, supplemental feed, deworming, and manure application. In terms of pathogens in the environment, we found that those features of rotational grazing seem to be beneficial for pathogen reduction in water and soil. And so we think the result is this reduced exposure to livestock. But at this point, we can't definitively say if this reduced exposure equals improved health. Some other factors that influence pathogen presence in water and soil were the paddock resting days, number of days in paddock, number of grazing days, stocking density, as well as rainfall and soil moisture. We also found that cattle drinking water sources far exceeded recommended guidelines for microbial water quality. I didn't show those results today, but I can answer questions about it. We also found pathogens in all cattle drinking water sources, including well water. In terms of recommendations, we think that perhaps reducing stocking density and increasing resting time may reduce pathogen presence and exposure. We believe manure application may be problematic, and therefore adequate composting time is important. So if producers have multiple composting piles, and make sure you're not putting fresh manure on land to reduce exposure to your livestock. We also think the treatment and testing of cattle drinking water sources might be prudent given the levels of contamination we saw. In terms of next steps, we still need to analyze our wildlife sample data and include it in the data set. We have been experimenting with some more complex modeling approaches to try to better tease out specific factors that are interrelated that should be optimized. And we really want to solicit feedback from producers and producer organizations to guide next steps for additional analyses. So that's partly why I'm here today. We did a longer webinar, so if folks are interested, I can send the link for that. And we're also presenting at a couple other symposiums. We also would like to investigate the links between water quality and cattle health after some different things were raised by the producers in our study that made us want to investigate that further. So we've submitted proposals for funding for that work. And so I'll finish there and acknowledge all our producers in the study, the funding provided by Beef Farmers of Ontario, the University of Guelph's Food from Thought Initiative, my Canada Research Chair, and the Canadian Foundation for Innovation, as well as all the students and staff from our lab that helped collect the samples over the different grazing seasons.